I think. All right, thanks for coming, everybody. We're gonna get started. And I need to stand over here, so just direct all your eyes this way, please. Actually, just as a point of business, before we get started, we are recording this session and webcasting it to other growers across Canada who wanted to join in. So that's why we're going to be standing over here. We've got the video camera here, and Lindsey will be presenting from here. So just so you know, and also to keep it as quiet as you can during the presentation. Rattle, rattle, rattle. It's fine. So I'm Alex Lyon. I'm a postdoctoral fellow at the Center for Sustainable Food Systems at UBC Farm, where I work with many collaborators, including Farm Folk, City Folk, and the Bauta Family Initiative on Canadian seed security, to do variety trials and participatory plant breeding with the CANOVI project. That's Canadian Organic Vegetable Improvement. And we are really delighted to be working with Lindsey du Toit. She is a vegetable seed pathologist from Washington State University, where she's been working for 19 years. And she co-leads the Pacific Northwest Vegetable Extension Group. And she runs an applied research program focusing on seed crops grown in the Pacific Northwest. I was Googling her and learned that she was recently awarded the Alfred Christensen Distinguished Professorship in Vegetable Seed Production, which I think is really cool that that is something that exists, and also shows the importance that her work has had for seed growers throughout the Pacific Northwest. I first started hearing about Lindsey when I began collaborating with Organic Seed Alliance, which also works in the Pacific Northwest and throughout the US with seed growers, and started learning through that, sort of hearing Lindsey's reputation as somebody who was working with the organic growers and helping them figure out issues with vegetable seed pathology. So when we started this project where we are going to be doing vegetable variety trials and participatory plant breeding, where we're potentially asking farmers to trade seeds with each other, we wanted to have some plans about how to manage the risk that might involve for transferring pathogens. And I was really thrilled that when I reached out to Lindsey, she was willing to work with us on this project, and also thrilled that she was able to come here today. So thank you so much for making the trip and for being here. I know when we were meeting with the BC Eco Seed Co-op yesterday, this was also something that came up, of the idea of what are our plans, what is our strategy as a community to manage these risks. And then another final point of business, I wanted to acknowledge that our presentation today is made possible by our funding from the Canadian Agricultural Partnership through the Organic Science Cluster 3. So that's the funding that we have for the CANOVI project. And also projects like this with funding from the Ministry of Agriculture require an industry partner to provide a third to up to half of the funding. And you're our industry. And we didn't want to go to any of you individually to ask for that much money. So we're really, really grateful that we have the Bauta Initiative and Seed Change to provide that industry funding. And they do so much more than that. They actually facilitate and have grown the networks that now allow us to have these variety trials throughout Canada. So very grateful for both of those entities. And with that, I'll turn it over to Lindsey. Oh, one final note, if you have quick questions to clarify things while Lindsey's talking, feel free to raise your hand. Tara's going to be typing those so people can see them online. If it's a longer question, please save it for the end, and then we'll have plenty of time for discussion. All right, thank you, Lindsey.
Thank you so much. Can you hear me at the back? I get accused of not having a loud enough voice, so if you hear my voice dropping off, just raise your hand or do this. I tell the students when I do this in the audience, they need to project, so it's my turn to be told that. So thank you very much, and it's wonderful that it's just an hour-and-a-half drive from my home to get here. It was very easy. I didn't have to get on a plane. That being said, most people don't want to go to the doctor when they hope they are healthy more than sick, and certainly as seed producers or seed savers or seed exchanges or seed stewards, you hope never to have to deal with people like me, because when I get called out to a seed crop, it's usually not for a good reason. So I've had people listen to some of these kinds of workshops we've done and walk out at the end and say, why do I bother raising seeds? You've given me nightmares. So it's not my intention, but really it is to help you be aware of some of the potential risks and what you can do to try and raise your consciousness, raise the consciousness of the folks with whom you're working, and just be aware that just like with human health, animal health, plants get sick too, and seeds can be bearers of those kinds of problems. The beauty of this kind of a group is that most of you are quite diversified in what you do, and that immediately reduces a significant percentage of risk compared to the larger scale growers with whom I work. So, when you get scale up operations, you immediately start to increase pressure and selection for these pathogens. So that's one of the things I've encountered in the groups that are smaller and more diversified. I tend to see far fewer of these catastrophes that can cause problems. But that doesn't mean you can let your guard down. And I think particularly because this industry is focused on sharing resources and exchanging resources, that's one risk that becomes of primary concern to try and address. So if you can keep that in mind as we go through this, hopefully you don't leave here with nightmares about everything that can go wrong. Okay. Anyone recognize, ignore the pathogens on the seed, but which seed these are? Obviously vegetables. I work on vegetables. I apologize if you don't work on vegetables, but that's my responsibility at Washington State University. So I'm vegetable centric. Yes. Okay. Which one's onion? Yes. Onion in the middle. And that's a fungus called botrytis or gray mold, which can be a problem in bulbs going into storage. It can be a problem in terms of the umbels on the seed crops and so on. Anyone recognize the left or the right? It's a bit hard because they've covered in pathogens that distorts their appearance. One on the left is a brassica seed, cabbage seed, infected with a fungus called Formalingum, causes a disease called blackleg. That is quite a serious concern if you're in the kind of climate we're in here in BC and Western Washington, Western Oregon, likes cool and wet. On the right-hand side is a spinach seed infected with a fungus called Stymphalium. Also likes wet, but it tends to like warm and wet. So we see big problems in places like California and Arizona, and Florida, and Texas, and so on. Okay, so we'll move on with that. Let's see if I can do this. So just to put everything in context of diseases, as a pathologist, we tend to be pathology-centric, and I have to remind myself and the folks with whom I work that it's just one of the many criteria that those of you working seed have to try to achieve. Obviously, if you're producing for a particular set of traits that you want to sell, whether it's the color or the shelf life or the flavor, the nutrition, those are you want that true to type genetically. And when you deal with cross-pollinated species, that becomes really important. Most of the vegetables with which I work are cross-pollinated, so a big part of helping seed growers is maintaining that genetic purity. If you're trying to select for certain traits, you don't want a red cabbage crossed with a white cabbage because now you're going to get a pink cabbage. And also, obviously, good quality seed in terms of germination and vigor. So if you exchange seed, you're hoping that the seed you give to someone or sell is going to have reasonable germination and vigor. And those are traits you're trying to produce as a seed producer. So pathology is just in the middle of all of these things that you have to consider. So again, don't come out of this thinking everything's about diseases, but as a pathologist, I tend to be that way. Also, another important trait is freedom from weed seed. So many of you know the difficulty of managing weeds, particularly in organic systems, where you've a climate like ours, where weeds can grow all winter. That complicates some of the ability to control weeds, and you're not wanting to disseminate weed seeds with yours. And of course, production costs. We've heard multiple comments this morning about the hope to make some money off your seed at some point so you can make a living doing seeds, but this can be difficult. These are intensive crops. They do require a significant amount of labor. And then pathology just becomes part of that as you talk about seed testing and the cost of seed testing, the resources needed to do good seed testing. It's not inexpensive, and it complicates this effort of trying to assure that your seeds are clean. And if at any point you need to ask questions, please do, because, so just to put in context, what are plant diseases? If you have a plant problem and you don't know what's causing your pumpkin to throw up, where do you begin? Part of knowing how to manage diseases is being aware of the diversity of types of things that can go wrong. And when a grower comes to me with a problem, they might think it's a disease because the plants are not looking good, but often it's not a disease. And a disease in the true sense, as a pathologist, is caused by an infectious organism. So a fungus, a bacterium, a virus, a nematode, or so on. But generally, people just lump everything into disease or pest. It could be insects, it could be abiotic. So when we're trying to help a grower, one of the important things is to figure out what's actually going on. Is it really a disease or is it something else? So we tend to divide diseases into these two categories of biotic, bio meaning living, so caused by a living organism, and abiotic, the problems associated with a non-living problem. So it could be environmental stress, it could be nutritional deficiency, or could be extremes in temperatures or soil moisture or something along those lines. And then within those biotic groups, we have three categories. This is where I focus as a pathologist, the infectious microbial types of organisms that can spread around. And we'll just speak a little bit about them. I'm not going to go into too much technical detail, but I think it really helps to understand why a bacterium is not a fungus, what that means in terms of management and sources of inoculum and how things move around and so on. We have obviously the entomology related area of insects and mites and so on. And we have what I like to call macros, so, the deer, the rodents, the other things that come in and feed on your crops and so on. And I know that we're not trying to manage them in the sense of get rid of them, but we are trying to live along with these systems and still produce seed. But I think a very important part that we often forget is these abiotic problems that can go wrong. So quite often I get called to look at a problem or a sample is brought to my lab and they think it's a disease, and we start doing more investigation, and it turns out it's really something non-pathogenic, which is good because it means non-infectious. Then the question is, how do you prevent that abiotic stress? Okay. But that's an extremely important distinction in managing. And so just to remind you about these different types of organisms, the pathogens, they're mostly microscopic. And that's why, unfortunately, you need pathologists. Just like you need in medicine, you need labs that can run tests for certain organisms, because you can't see them most of the time with the naked eye, sometimes not even with a microscope, depending on the size of these organisms. And they reproduce in different ways. They survive in different ways. They infect in different ways. And I think these are, you don't have to know all that information, but being aware that not all pathogens behave the same way can help you be aware of the need to, if you do have a problem and you do find out what it is, understanding a little bit about how that organism operates gives you a clue to where you might be able to manage it, where you're not going to be able to manage it, and what your options are, okay? So, there's a drawing, it's a famous drawing from Agrios. He's a deceased plant pathologist from Florida, has a wonderful introductory textbook if anyone wants to. So good bedtime reading, but this is a plant cell, you can see on the overall, and just to give you an idea of scale, the very top is the head of a nematode. And so nematodes have this little stylet in their mouth, and it's quite vicious, and they probe it in and out very fast, and it damages the cells, and they suck up the contents of the cells through that stylet. You can see it inside the head. And they can be up to, about a couple millimeters long. They're very, very big ones. So I don't know if you're okay if I'm metric or non-metric. I'm not sure what people are in Canada. It's everything. So they're tiny, but they're big in the worlds of pathogens. So some of them you can almost see with the naked eye. And then you can see what's the fungus. Maybe if I use the cursor, would that help? See if does it show? Nope. Okay. Slide it over to the right. Sorry, I'm not quite sure how to do. I'll just use my language. So you can see the word fungus over there. That's so what's called a hyphum. It's protruding into the cell and extruding enzymes and breaking down the cell and feeding. You can see a bacterium. Bacterium is a single cell. And a single cell can divide within 20 minutes. And so every 20 minutes, potentially, a bacterium population can double. So you can see within a day, your population go from 1 or 2 cells on your plant to an extraordinary number if you into math, okay? And bacteria reproduce extremely fast. They're very simple. They're favored typically by wet conditions because they splash disperse most of the time. Some of them are insect vectored, but you can see the scale. And then we have this group of things called, they call them molecules here, they're also called phytoplasmas. They're like bacteria, they don't have a wall around them. They're carried by leafhoppers. And when the leafhoppers feed, they inject them into the phloem, and they move up and down the phloem. So they have these odd characteristics that they cause on the host because of the interference in the way the food moves up and down the plant. So, and then you can see a bunch of viruses just listed there. The words are bigger than the size of those viruses, and viroids get even smaller. Viroids are just a piece of There's no protein around them, just a piece of RNA or DNA, whereas viruses actually have a piece of RNA or DNA with a protein shell around them. Now, this gets to you think, why am I telling you all this stuff? But in the end, it does become important in understanding how these things reproduce and move around and so on. So I won't go into details because there's thousands and hundreds of thousands of different examples. But what you're trying to get to as a producer or as a seeds person is understanding how that pathogen life cycle fits into your seed cycle. Okay. So these pathogens, because they're living organisms, they have to somehow get onto your plant. How do they get onto the plant? Is it rain? Is it you handling infected material and going over and, sorry, I'm supposed to stand in front of the camera, going over and touching another plant and spreading it? Some things are spread by hand, some are not. Is it your pruning shears if you're cutting or you're harvesting things? Different organisms spread in different ways. So these are important things to think about. So they have to actually land on the host, and then they have to germinate like a seed does. The spore or the bacterial cell or whatever it is has to grow and reproduce and colonize, and then get into the host and reproduce. And you get the cycle going on. And there's different things that are needed for that. So some organisms, particularly fungi and bacteria, they need moisture. Most of them are favored by wet conditions. And that's why so much seed production is in the Western North American area, because we tend to have dry summers. When the seed is maturing, that's one of the most important traits for healthy seed, is to keep that seed dry. Okay. That's why you can do seed production in the Midwest, the East Coast, but the high humidity and the frequent rains that occur tend to have much, much higher risk of seed-borne pathogens. So most seed production, and at least on the commercial side, almost all of it happens in the western parts of North America, okay, because of moisture. So any questions related to this? The survival part is really, really important. So knowing how these things, that if they show up on your farm or in your crop, knowing how that organism, is it going to survive in your area? How is it going to survive? And what are your options for interfering in that life cycle? So when I start to look at a disease and a particular seed farm is experiencing, one of the things we talk about is this life cycle of the pathogen and the life cycle of the host. And where's the Achilles heel in the pathogen? There are some pathogens where there's certain stages in the life you may as well not even begin to think about, because you're not going to disrupt that part of the life cycle. There are other stages that are there's a real vulnerability in your ability to come in and disrupt that life cycle. So if you can think about it that way, when you do encounter a disease, it can make a big difference in your ability to get that disease in check, okay? Or prevent it from happening in the first place. Does that make sense? I mean, it's all logical, but sometimes we just need to be reminded, okay? And so when you think about seed crops, particularly crops that are, say, biennial or perennial, or you start getting into lots of periods of risk. So let's just use cabbage as an example, or brassicas, biennial brassicas. You know, the stock seed is planted usually in greenhouses or hoop houses, and you produce the transplants. You plant them out in this area. We plant them out in August, September. They have to go through the winter. They have to vernalize. You might have to cover them with remade so they don't freeze out when it gets down to minus 10 degrees Celsius. Whenever you cover things like that, now you start to warm up the area around the plants. You also increase the humidity. So we start seeing diseases like downy mildews and some of these other fungal and bacterial pathogens potentially becoming a problem under that remade if you don't get it off early enough. Or if you let it sit around and suddenly it warms up to 10, 15 degrees Celsius, and then you know it's going to freeze the next week and you don't take it off, you're favoring this environment that's very suitable for certain pathogens. And then, of course, you know that when things bolt in the spring and seed stalks come out, there's a lot of soft, succulent, rapid-growing tissue. You might get a hard, late freeze in the spring. That physical damage can encourage things like gray mold, the botrytis species that are quite prevalent in our area. So we start to see those problems show up. All these periods where conditions can fluctuate between favorable and unfavorable for different things. And of course, during flowering, you have a big canopy. So what happens when you have a big canopy? What does that mean? So you start getting restricted airflow. You start getting increase in humidity, and we could still have rains during, you know, June May when these are flowering. And so that big canopy, restricted airflow can make a big difference in favorable conditions for diseases. And of course, then in the case of some of these species, they have to be pollinated. There are a few pathogens that are carried on pollen, unfortunately. You know, blueberry growers know that very well, blueberry shock virus and others. Fortunately, most are not, but there's a few crops where you have to be aware of this. And of course, in the case of many of the naked seed vegetables with which I work, the seed has to go through a drying period. The crop has to dry down, senesce, the seed has to mature. If it rains when your crop is windrowed or drying out there in the field, that's one of your most dangerous periods. And this is why, again, I said most seed production's in the western part of North America, because we tend to be dry during the summer and dry when the seed should be maturing. In places like Denmark, where they don't, which is a big seed production area for some species, they don't necessarily have a guaranteed dry July, August, like we tend to here on the west side of the Cascades. So they actually have to dry indoors. So they're set up, most of the farms, the seed farms, have their own dryers because they know they have to if the weather doesn't cooperate. And it's all about diseases and seed quality. Okay. So diseases can affect your seed production in multiple ways. The obvious one is that you get less seed. If it's severe enough and it's affecting growth and your crops are smaller, your plants are shorter, or just not photosynthesized enough, you might get a reduction in yield. But it could be you get normal production, but you have quality reduction. Some pathogens will affect seed germination, seed vigor. Now it's going to be harder to sell your seed if your germination goes from 85% down to 50%. You're not going to want to buy a seed lot that's at 50% if you're one of those recipients of that seed. Others don't affect seed quality at all, but they do pose a risk because now you have the potential to move these things around on your seed. And that's obviously when we talk about seed exchange movements like this, where you're about sharing resources, this is one of the key things you want to think about. Okay. Any questions on this? It's all pretty obvious.
Well, it's like I'm not sure, but are there particular diseases that we do concerned about spreading?
Yeah, we'll go through some examples. So the question is, are there some particular diseases that you're most concerned about being on seed? And absolutely. Some things, not all pathogens are seed-borne. That's an extremely important issue to remember. You may have a disease show up in your seed crop, and it may be quite devastating, but it has no risk of being seed-borne. So any seed you do harvest is still not a risk. So it's important to know if the particular pathogen or disease you're encountering is seed-borne or not. And so this is a critical part of being a seed pathologist, is helping growers know if there's a risk or not. So there's a virus that's a very serious problem in onion seed production east of the Cascades. One of you, I think, said you grow onions and carrots east of the Cascades, and it can be very, very serious. It's thrips vected, and it can, I've seen seed crops completely wiped out multiple times, 2 weeks before harvest. But any seed you get off those crops is not infected. So if you take that seed off, you have to clean it and size it. You lose 80% of it because of, the sizing doesn't finish off. But what does make size is perfectly fine and not a risk for seed-borne iris yellow spot virus. So, knowing these things is really helpful. If you're a seed grower and you have this wipeout, do I have to get rid of the seed, or can I still harvest that seed? Okay, so that's an indirect answer to your question, but very important. Okay. So how many of you, hopefully, have heard of the disease triangle? Or actually, I say, who has not heard of the disease triangle? And it's okay to raise your hand. It's important. Because this is a fundamental principle. Have not heard the disease triangle.
Or the disease triangle.
Yeah, sorry. If I'm speaking too fast, just, can you hear me at the back still? Okay. So disease triangle is a very simple concept, but it's solid. It works. It works all the time. It's one of those few things that doesn't have exceptions. And the concept here is the triangle represents the amount of disease. The bigger the triangle, the more disease. The smaller the triangle, less disease. There are three things you have to have disease. And the amount of each of those three things dictates the size of that triangle, how much disease you have. So you have to have the pathogen. No pathogen, no risk. Okay. You have to have the host. No host, no risk. Very obvious. You also have to have a favorable environment. So the more favorable environment for that pathogen, the bigger that side of the triangle is. So the length of each side of that triangle is determined by the amount of that factor. So if you've got a big monocropping type system, you've got a long bottom of that triangle, the triangle is going to be even bigger in terms of potential disease risk. If you have a lot of pathogen present, because it's an endemic disease and it's been building up and there's no crop rotation and all these things that we tell you not to do, that side of the triangle is going to be huge. So when you think about a disease that might show up on your farm, how do you manage it? So this is a very fundamental concept that we talk about in plant pathology with our students. Is there one or more sides of this triangle that you can reduce in size to get a smaller triangle? You may not be able to eradicate the disease, but can you reduce the size of the triangle? And it's simple, it seems obvious, but we over and over again, we forget this concept. And if we go back to this concept, it helps you think systematically through the process of how you might manage a problem that shows up on your farm. Do you have the capacity to reduce the amount of pathogen? How do you think that might happen? We're going to talk about it, but just see if anyone can think of anything. If you know you have a disease on your farm.
Well, prior irrigation, just basically
So
do your.
if you know where the pathogen is surviving, how it's persisting, are there ways that you can interrupt that? Okay. Yeah. So sanitation, roguing out infected plants is a classic way of reducing the amount of inoculum. Yep. How about changing the environment? Shortening that side of the triangle. So we're going to talk about examples of all of this. So this is a very fundamental principle, and it helps to think about this when you do encounter a situation. How about the host? How might you reduce, without cutting down the size? You might have, say, a 20-foot by 50-foot plot of whatever it is you're growing. You don't want to reduce the amount of seed you're producing, but how might you alter the host susceptibility to the disease? So selecting varieties or parent lines that have better resistance is an example. Yep, exactly. Or healthier plants. Yep, less stressed plants. Yep, exactly. Okay, so let's start going through some of these practices. So I'm going to focus initially on cultural practices, and this is obviously particularly important if you're doing organic seed production. I work with everything out there under the sun in terms of what producers do, from your hard nozzle heads to your tree huggers and everything in between, which is wonderful as a scientist because I'm constantly challenged about how do I present options that fit that person's philosophy. As a public scientist, it's not my right to tell people how they produce, but my job is to help provide them with management practices that meet their philosophy and that are legal. So I mentioned location is really important. Now, this is hard if you've got your 2 acres somewhere and you bought it, you're not moving, okay? But this is exactly why seed production happens in this part of the world within North America, because we have favorable summers. But this is a very fundamental principle. So continentally, areas that are drier during seed development are quite critical. Even in, say, Washington State, where I work, we have these pockets in Western Washington of drier climates. So the Sequim, Clallam County over on Vancouver, the North Olympic Peninsula, that area gets half the rain that Mount Vernon does because it's in a rain shadow from the Olympic Peninsula. So it's a very popular seed production region. Even though it's on the rainy side of the Cascades, it's a rain shadow area. It gets significant, less than half of what we get in Mount Vernon, which is only about 50 miles away as a crow flies. Well, less than that, actually. Okay. So these are very fundamental principles and it's all about disease management and quality of seed, okay? So locally, what can you do? And you can think about things like avoiding areas where you might have frost pockets, if you've got cold sensitivity, or areas that are prone to fogs or dews, or areas where you have restricted airflow. If you have a big bank of trees on one side of your farm and you know there's a predominant wind direction, it can make a big difference by thinking about where you locate your crop and how you orient your crop to take advantage of better airflow, better movement of the wind through your crop so that it dries down faster after it rains or after you've irrigated, okay? This becomes a really big one, and maybe less so for small diversified operations, but this thing called the Green Bridge. How many of you have heard of the Green Bridge? Okay. This is an extremely important principle, particularly when you get to seed crops that have overlapping seasons, so biennial or perennial, because one season of a seed crop overlaps with the next. So if one crop is diseased or has an insect pest and the next crop is now being planted and started growing, as this current crop dries down, if it's something that can move like insects, they're going to fly over to the next crop. So when you don't have that spatial separation and temporal separation, that Green Bridge effect really lends itself to propagating these pathogens in seed production. And it's one of the reasons that for certain plant species, you'll find the seed industry tries to separate itself from the primary production regions. because of this, not keeping the seed away from these larger scales of production to break that green bridge. Okay. Now, most of you are dealing with fairly diversified operations. That's immediately a huge advantage. Okay. But think about it, because there's some extremely important examples that are very relevant to diversified operations, and I'll give you some cases to go through. So where I work in Washington is just south of the border. Sorry, it's just explaining how we have two key areas of seed production highlighted in red. I'm based at the Mount Vernon Research Center point in the top corner, so not too far from here. But these are two key pockets for seed production because of the suitability of these climates, okay? And seed production in the Pacific Northwest, Oregon, Washington, Idaho, coming into Canada, produces, so for example, 75% of the world's carrot seed is grown in Central Oregon and Central Washington. Now, if you're into local seed, that's a horrifying fact, but that's a reality of the global seed system, okay? 75% of the world's carrot seed is grown in 2 states. Okay, just to put things in perspective. Just saying. So when there's a disaster, you can imagine the impact it has, right? This is why I have a job, unfortunately. One of the crops that, talking about carrots, one of the, to illustrate this green bridge effect and this issue of isolation, bacterial blight of carrots is a very serious issue, not because it causes huge epidemics in the crops, but because it's very readily seed-borne. And so you can see what it does to the umbels. It's a bacterium called Xanthomonas. It's very readily infects the umbels, and it very rarely becomes seed-borne, and now a high risk of seed transmission if you sell that seed. So we're trying to figure out, this disease has become endemic in Central Oregon and Central Washington, particularly Central Oregon. So when I was first hired in 2000, the growers are saying, why is, we don't see the disease that much in these crops. Why do we see it so much on our seed? And so we started doing a bunch of survey work. And one of the things we did is we drove behind a combine that was harvesting. So obviously this is big scale, but just the principle here is what you need to listen to. And we had this little air sampler that we put on the back of the pickup and drove behind these combines at varying distances. And we were able to trap Xanthomonas in the airborne aerosolized residues, the dust coming out from those combines, up to a mile downwind of the combine. Now, this is a biennial crop. There's about a 6 to 8 week overlap from one season to the next in the cropping. So when they're harvesting, all of next year's crops are in the ground and up out of the ground. Nice young succulent plants, overhead irrigated, perfect for bacteria. This is why Xanthomonas become endemic in our area. We don't see symptoms. This bacterium colonizes the leaves asymptomatically. And it will increase in population until you get to close to 1 million bacteria per gram of leaf tissue before you see symptoms. So by the time you see symptoms, there's no way to control it. So it's complicated, and this is the difficulty of managing these diseases. So, but again, this is larger scale, intensified areas of concentrated seed crops. They isolated space-wise for pollen because they have to, it's cross-pollinated species, but not temporally. And this lack of breaking the green bridge has been a huge part of why we have a big problem with this disease now. So if you're isolated in your carrot seed crops in the inland BC, you should be pretty safe. It's one of the best things possible. Do you have a lot of carrot seed production in your area?
Not much. There's a little bit. Yeah.
Okay. Okay. But Queen Anne's lace. Yeah. Okay. Well, good. You're not concentrated. You had a question?
I was just going to ask, does this host live on Queen Anne's lace?
Yes, unfortunately, it does go to Queen Anne's lace. It tends not to cause nearly the same amount of population increase. It primarily increases and causes problems on carrots, but it can survive and persist to a small degree on some of the other APAC members. Yeah. Okay. So a great example of breaking this green bridge is a system that was implemented in Washington State in the 1940s. So around the end of World War II, beet seed production, table beet seed production, has been happening in Washington State and Oregon for over 100 years because the climate is so suitable on this mild maritime region. But after World War II, the beet seed yields had plummeted to about 1/10 of what they used to be, and the seed growers were very concerned about why this was happening. So they had a pathologist come out from Wisconsin to look at this, and after surveying, he realized that there were some viruses that were carried by aphids. Okay, you all know what aphids are. They fly. And these viruses were being moved by aphids in that green bridge from one season's crop to the next season's crop in that biennial system. Table beets overlap from one season to the next by 3 months. There's lots of opportunity when that seed crop is senescing and dying down. Those aphids are very mobile. They fly off and we're carrying these viruses with them. And so as a result of this, this is just showing the table beet seed production through the winter, and then they transplant them out in the spring. There's a seed crop being harvested. But this is what the virus can do to these crops and have a significant impact on yield, even though they're not seed-borne. So you don't have to worry about selling that seed, but if your seed yield is 1/10 of what it was, that's not good. So now the entire beet seed industry, and again, these are sort of commercial scale operations, they all plant their stock seed on Whidbey Island in July for the next year's crop. Every company, every grower has to have their stock seed planted on Whidbey Island. So there's no beets on the mainland all winter. In the spring, those beets get dug up and transplanted on the mainland. They've broken the green bridge. This system has been in place since the 1940s. We do not have problems with these viruses. No chemicals needed to spray for aphids. Breaking the green bridge. But it's a big cost. It's a big logistical pain. About every 10 years, we have new players in the industry who say, why do we do this? We're wasting our money and time, and let's just go back to the old system and have to bring out the history books and talk about the green bridge on the scale that they're growing these crops. Breaking this green bridge is really important. We don't want to have to start spraying a ton of insecticides that don't work well against the way that these viruses are transmitted by the aphids. So we have to go back to basic biology and repeat this lesson about every 10 years. Okay. Yeah.
What if you have a bad year on it?
Well, that's an issue. Do you have a bad year on Whidbey? So what? Yeah. So one of the things that companies do is they grow these crops around the world. So many of these companies that produce in Washington, Oregon, also produce in New Zealand, in South Africa, in Chile, in Denmark. So they need to be away from the equator to have that enough of a chilling factor, but not cold enough to kill. So they spread their risk. Again, this is a global system. So now we're moving seed all over the world, potentially also with pathogens. Yep.
So are they not allowed to do any seed crop on the beet?
Yes, so most of them are not growing. You'll see a few crops on Whidbey, but they're isolated from the key areas where the beds are planted. Yeah, yeah. And this is voluntary, but the industry has been burned so badly by this disease in the past. And again, this is larger scale, but think about this on your scale of operation, this breaking of the green bridge.
So there's one farm on Whidbey that hosts everybody?
There's about 3 farms that rent their land out to everyone in the winter, or basically from mid-June, they make the beds and then they harvest in March, April. Yeah. So it's giving those farmers on Whidbey a chance to rent land and get income as well. Yeah.
What percentage of that area
from this area? It fluctuates, but between Washington and Oregon, we're probably producing, I'm guessing, 40% of the world's beet seed.
So on duplicating that whole concept on a micro scale, we could create little isolated areas within our region that could serve the same way as the island.
Yeah, if we
have identified this.
If you're diversified, and most of you are, at least I'm making assumptions here based on working with groups like this over the years, that most of your operations tend to be growing not just 1 or 2 species. And not typically, you're not surrounded by 20 other farms doing the same 3 or 4 crops. That's what increases this risk because of that selection pressure for that pathogen. So you might not have quite the same need for this extreme sense of isolation. But I'll give you some examples where the story changes for small scales, and we'll go through some examples here. But I think there's some valuable lessons here to think about. All right, so this is the example I want you to think about. You led me into my next slide, thank you. So the crops here on this slide, I took this photo just over a year ago. This is in Skagit County, Washington State. These are all brassicas. Different brassicas, but they're all brassicas. This particular farm grows brassicas year-round for fresh market for different reasons. It's a CSA farmer's market operation, and you're harvesting year round. Unfortunately, there are a few diseases that are not just species specific, but family specific. So black rot, which is another Xanthomonas, it's a different Xanthomonas than the one that goes to carrots, but behaves quite similarly, and also seed-borne, and very, very low tolerance for this pathogen. In fact, if Xanthomonas campestris is detected on a brassica seed lot, in the commercial world, it cannot be sold. It has to be hot water treated, retested, and only sold if it tests negative after treatment. Okay, hot water treatment can work quite well, but it's very explosive under typical brassica production practices. Most people grow brassicas with overhead irrigation. This is very, very severely splash dispersed. Okay, so this is particular. This was the sample the grower brought to my lab, and that you can see the symptoms starting to show up. It's actually a quarantine disease in six counties in Washington State that are key seed production region for brassica crops, because we don't want the disease contaminating the seed production area. Unfortunately, this farm was in the seed production region. So this farmer was required to destroy that crop by law because of the risk of that disease establishing. This pathogen will go to any brassica weed. So those of you who know brassicas and know weeds in this area, BC included, there's a ton of brassica weeds that grow all winter. So the risk of this disease establishing, because it can move to all these brassica weeds, is so high that the seed growers requested this disease be established as a quarantine pathogen for our area. So any crop that comes up positive has to be destroyed. This past spring, we had 5 cabbage seed crops have to be destroyed over another pathogen, the blackleg pathogen, Formalingum, that also goes to brassicas, because 5 crops showed up positive in March and April this spring. They all had to be destroyed. Growers never got paid very much. They got reimbursed for the expenses to that date. The seed companies had no money to show, no seed to show for it, and they had to pay out money. So you can see that when you get to these pathogens that have a host range that now becomes a problem, even though you're diversified, many diversified farms in this region do a lot of different brassicas. So this is where the story about isolation becomes important for your area. And this is probably black rot, and more so blackleg is the one disease I worry about with some of the seed groups exchanging in this area, because we have such a favorable climate for blackleg. Now, I'm not trying to create a horror story and nightmares, but consciousness of, if you see a problem and you're not sure what it is, get it diagnosed. Get someone to look at it and see if you have it, because you don't want to be spreading these pathogens to your friends, the folks we're trying to support. Okay. The good thing is, so this is the blackleg one, so it's Formalingum, also brassicas. This is just like the black rot, goes to just about every brassica out there. These 2 pathogens are not soil-borne, okay? They will only persist in soil on residues of infected plant material. So the good thing is they're not going to sit in that soil for 10 years. If you incorporate the residues and break them down, those pathogens will disappear if there aren't hosts around. The pathogens that are soil-borne are the really problematic ones, because they're persistent. So Formalingum and Xanthomonas campestris don't survive in soil in the absence of host tissue. But if you leave those residues on the soil surface, they will sit around as long as those residues are still there. And that's one of the reasons we talk about, if you suspect you have a problem, we know that no tillage and good ground cover is an important principle of healthy soil management. But if you know you've got infected material, one of the best things to do for these ones that are not soil-borne is to get that material broken up and incorporated into the soil. Let the soil microbes break things down fast. They won't break down very fast on the soil surface, and you can get rid of your inoculum much quicker. If you've got these big, fat stems from, broccoli or cabbage seed crops, those are going to take a while to break down. So breaking up that residue as much as you can before you incorporate it will speed up the inoculum disappearing. Okay, does that help that question? So I bring this up because in blackleg, caused by Formalingum, was considered, in the 1970s, there was an outbreak of blackleg in cabbage crops in New York and the Midwest. And all of those outbreaks were tied back to seed grown here in Western Washington, Western Oregon. And it had devastating impacts on seed production for brassicas. So the industry learned a very hard lesson about good practices in keeping these diseases out, because we have such a favorable climate for these biennials. And unfortunately, this disease was considered to be in check for a long time, but in 2014, it showed up all across the Willamette Valley. in a very short time. So Cynthia Oakham, who's a pathologist at Oregon State University, was surveying out there, and these are just the wide range of different brassicas that she found blackleg on in 2014. And she'd been surveying in that area for years, so it wasn't like she just hadn't noticed it. And so then the question is, where on earth did all this inoculum come from in such short order in the Willamette Valley? It was in organic farms, it was in conventional farms, it was on these larger canola crops, it was on weeds along the roadsides, and it was quite disconcerting. This is a picture taken in an organic farm that does both seed production and fresh market. And you can see the loss in the kale from blackleg. This picture was taken in January 2015. This is a picture of Cindy Oakham in the brown sweater. Shannon was a graduate student in my program in the greens raincoat, and we were standing in a canola crop in the Willamette Valley, rainy and cold. It's February, that's February. But if you look at the canola crops, I'm not sure how well you can see, but all those lesions on the leaves and all the little black specks inside those lesions are the fruiting bodies oozing out their spores, because when they imbibe moisture from the rain, they basically push their spores out like squeezing a toothpaste tube, and then they get splashed around. So this canola field had 100% incidence of blackleg. 100% incidence. So this is the winter after that disease showed up in the Willamette Valley. And it really sort of took all of us off guard. How did this happen? And I won't get into the details, and I'm happy to chat over lunch about this because it's a sordid story, but it made us so cognizant of the importance of not letting your guard down. Even diseases that we thought we had in check, if things start to fall apart, we forget things because we haven't seen the disease in 30 years, and suddenly things can change if we're not careful. So it was a very good lesson for all of us. So I mentioned something about this. Yeah, question there?
Blackleg, do you know where it is now?
It stops at the Oregon border. Doesn't have a passport. Unfortunately, it has, as I said, it showed up in Washington. We hadn't seen it in Washington since the 70s, and it showed up in 5 fields last spring with required crop destruction. It was devastating. So we don't know when it showed up in Washington whether or not the companies went back and checked their records. Their stock seed lots had all tested negative. We're actually setting up spore traps this winter to monitor in the area to see if it has established in Western Washington. We didn't see it show up in any other fields after those crops were destroyed. What I suspect happened is these growers were getting their transplants. Most of them came from a nursery in California. And that nursery produces transplants for hundreds of operations around the place. And not all of them are worried about blackleg. So not all of them are making sure that the seed going into that nursery is clean. And I suspect that somewhere in that nursery cross-contamination happened. Transplants got shipped to Washington, and yeah. So this is, when you move stuff around, we start to have these risks. And the companies that had produced those seedlings in California previously were using a nursery in Oregon and had switched nurseries for various reasons. So anyway, it's a good lesson, yeah.
I was thinking about breast earlier. Do you see evolution variation breeding for blackleg or
Yeah, we do, as we've been collecting isolates of this fungus from Oregon and Washington, we've been working with a pathologist at the University of Manitoba who, blacklegs is the #1 problem for canola production in Canada, for, because of the scale of production. Talk about monoculture, it's huge. And they're constantly breeding for resistance to blackleg, and blackleg fungus adjusts. So you've got all these races. It's a race war in the world of canola, of breeding for resistance, the fungus overcomes it. Breeding for, it's a boom bust war constantly. And you know that more than I do. So this pathologist in Manitoba is very familiar with all the races. And so he tested the races we were picking up in Oregon. We found a brand new race that had not been found anywhere in the world before. This fungus reproduces sexually and asexually. And the significance of that, that's actually the reason I put that top picture in there. I wasn't going to go into it for time's sake, but this fungus, when it goes into its survival state on residues, crop residues, so that's the stem, bottom of the stem piece of a canola plant, you can see all those little bumps on there. Those are little fruiting bodies of the sexual stage of the fungus. And why that's significant is when it's going through its sexual stage, it's recombining genetically, which means more diversity in the pathogen, which means when you're planting resistance, there's a possibility that some of that genetically diverse population can overcome that resistance. And this is why it's a constant race. And in Oregon, the sexual spores also get shot into the air. The spores that you see on that leaf are the asexual spores. They splashed. So that's why rain and overhead irrigation favor them. The very bad part about this fungus is the sexual stage, because they're shot into the air like little cannonballs and they get picked up on the wind, and they can be blown miles. So now instead of splashing in a field, you've got, in fact, Cindy Oakham, the way she started monitoring this disease is she would set these shepherd's crooks up on the borders of fields with cabbage plants in these little pots, leave them out there for a week, bring them back into the greenhouse, and lo and behold, blackleg was developing. So these are aerially dispersed spores of the sexual stage that are moving between fields now. Okay.
Does that essentially mean if it's jumping in weed crops or like native brassicas, that there's no way?
Well, that's the problem. The cat's out of the bag in Oregon. We're not getting this disease back in check now because we know it's moved into, they've been monitoring weeds along I-5. It's establishing quite effectively. And that's why we're concerned when it showed up in Washington this past spring. Is it just a one-time introduction? The crops had to be destroyed straight away. We're hoping that we nip that one in the bud before the sexual stage could start forming. We're monitoring, we're setting out these traps. We really hope we succeeded, but yeah. And I don't want you to walk out of here horrified of brassicas and blackleg, but this is a reality. It's just like with human medicine and human diseases and movements of pathogens. And when we live in concentrations and populations, that's why we do vaccinations. That's why we do all these, things that I don't want to get into politics, but this is the reality of pathogens are living things and they move around. Okay. So I mentioned this virus that goes to onions. So this is a picture was taken by one of our extension agents at the time. So again, this is big scale, but trying to think about this idea of dissemination. So this is a virus called Iris yellow spot virus, IYSV, and it's carried by thrips. It's carried by one particular species of thrips, the onion thrips, not Western flower thrips, not the other thrips. But onion thrips are extremely common in our area, and they love onions. And so this is a picture taken in central Washington of an onion bulb crop. And it was actually, it's under a pivot, but they did it by drip irrigation. And you can see the headlines of the drip tape. And it's an infrared photo. So red is healthy, okay? Brown is unhealthy. And on the right-hand side is a smaller field that was planted to an onion seed crop. And that onion seed crop succumbed to Iris yellow spot virus. And when it was drying down in July, because that's when onion seed crops mature in our area, where did those thrips go? They just flew over to the bulb crop. So you can see that gradation of disease severity to die back in that onion bulb crop because of the thrips moving over. Thrips, adult thrips fly. The young ones don't have wings yet, so they just crawl. But when that crop was drying down, the adults said, okay, we're out of here. We move over to the next thing to feed on. And we've seen this green bridge effect over and over again when you've got annual bulb crops and biennial seed crops. So if you're an onion grower, do you grow onions? You said a few. If you're doing onion bulb crops and onion seed crops, or if you're doing carrot root crops and carrot seed crops, this is where you start worrying because it's increasing that green bridge. Okay. So this is just a classic example of a severe form of green bridge. And this is a picture taken in Central Oregon. You can see the dieback on the left side of that seed crop. The field that's bare right now was last year's seed crop. Yep, where do the thrips move? They just fly over. And again, that overlaps. So a classic case that we see many times, insect vectored. Obviously, when things can fly, that makes it worse. All right, so isolation. We all have that principle in our heads now. So let's talk about rotation. It's kind of along the same lines. Crop rotation makes sense. You don't plant the same thing on the same piece of ground over and over again, although I see that all the time, for nutritional reasons, for various reasons, but one of them is also diseases. And particularly, crop rotation can be extremely valuable for managing diseases. If the diseases are not soil-borne, then rotation is extremely effective because there's a limited time that things will survive in that soil. If it's soil-borne, then rotation is still important, it's still valuable, but it's not going to have as big an impact on knocking back your pathogen population because these soil-borne things can keep going in that soil for a while. So these are the duration that you have to rotate out of a field is influenced by all of these things. That's the main point of this slide. Okay. And also, as I mentioned, some things can be asymptomatic hosts, and that's a problem if you don't know it's there because you can't see the symptoms, but potentially it's your pathogen of concern could be surviving on these asymptomatic hosts. And then just remember that there can be alternative hosts like weeds for some of these pathogens. So how do you get rid of inoculum? How do you reduce the pathogen side of that triangle, get that side of the triangle short? You know, you can remove infected debris. Someone talked about roguing out infected symptomatic plants that you're suspicious of. There's ways to reduce inoculum of soil-borne pathogens or suppress that inoculum. We'll talk about that. Historically, people did things like burning residues. And one of the biggest reasons people burnt residues was to kill pathogens and insects. Now, burning has ecological issues. It has, you know, environmental and other issues. So it's being done less and less, but that was the primary reason burning was used, was disease and pest management. You can get into things like biofumigation, you know, using these biofumigant cover crops like mustards high in glucosinolates, that when you incorporate them, they release a naturally occurring biofumigant that helps knock back populations. Used very, very widely. Central Washington, on average year, there's about 50,000 acres of mustard biofumigant crops to help control some of the soil-borne pathogens that affect potatoes. It's a big potato production area. This is on a commercial scale, but it's a very good alternative to synthetic fumigants. Okay. Soil cellularization can be very effective in hot, dry climates. We don't have that kind of environment here. You talk about Israel and Arizona, those kinds of regions. And I'm going to give you a few illustrations. So these are two foliar pathogens that we know affect spinach production in our area. They cause leaf spots. We've shown through research that Cladosporium, one of the two leaf spot pathogens, survives on volunteer spinach. So when you harvest a spinach seed crop, usually some of that seed shatters and falls to the ground. Okay, you try and keep as much of it, but it's impossible not to have some of it fall to the ground. Spinach does very well in our winters. It will grow all winter. I mean, you've seen it growing out there. Unfortunately, Cladosporium likes cold too. So we found Cladosporium surviving on the volunteers after harvest of spinach seed crops because this fungus likes cool and spinach will grow through the winter. So we've talked about the importance of our growers when they've done harvesting, if they see those volunteers coming in, incorporating those volunteers back into the ground so that this pathogen is not continuing to persist. The other fungus, Stemphylium, we never found it on the winter volunteers because it likes warmer temperatures. It doesn't do well in the cold winter temperatures we have. But what we did find is you see the stem pieces as almost like woody-like tissue from the stems. They have those little black specks on there. Those are the sexual fruiting bodies, similar to the blackleg fungus forming those sexual fruiting bodies. And they have to go through a cold period to vernalize, just like many plants have to vernalize, some fungi have to vernalize. And in the spring, there's the little fruiting bodies. They produce these spores that are shot out into the air, so very similar to the way the sexual spores of blackleg are spread out. And we showed that if you leave your spinach residues on the surface of the ground, the sexual fruiting bodies will form, and in the spring they release their spores. And we showed that those residues can release spores for up to 2 years. We set up a cemetery of spinach residues. We had little plots where we had residues, you know, in mesh bags on the surface of the soil or buried. And every 5 weeks for 2 years, we'd go back and sample and put them in the lab, and we just invert a Petri plate over the residues, and the spores would get shot out, and we'd be able to trap them and quantify how much spore release was happening. And we picked up spore release for up to 2 years. If you bury them, nothing happens. The residues won't form the sexual stage, it won't vert the sexual bodies. If they do form, won't vernalize, and it can't release spores. So burying those residues, it's kind of like burning, but you're not environmentally destructive. So a really effective way of managing both of these pathogens is to incorporate residues after harvest. Simple. Okay. We talked about biofumigant cover crops. These can be really effective for reducing some of the soil-borne inoculum. One of the diseases that we have a big problem with in spinach seed production. How many of you do spinach seed? Anyone? Yeah. Okay, a few of you do. All right. So spinach seed does very well in our mild climate. It's an annual, not a biennial. It doesn't devernalize. It bolts in response to day length. Unfortunately, there's a fungus that attacks spinach called Fusarium wilt, or the disease is called Fusarium wilt, that really likes the acid soils of our region. And it's extremely problematic. So we've done a whole series of trials over the years looking at ways we can put calcium carbonate in the soil, so naturally occurring limestone in the soil to raise the pH. And we did these trials, and this is just showing as we increase the rate of limestone amendment in the soil, calcium carbonate, the seed yield in these crops, okay? And look at the consistent effect that as you increase the pH and increase the calcium through calcium carbonate, you get an increase in seed yield, and it varies depending on the susceptibility of the parent line. What happened with that black line? You see that black line, how that particular female line, we went from 1,300 pounds of seed per acre to 2,500, and then it just plummeted as we continued to increase the rate of limestone amendment. Why on earth do you think that happened? We weren't anticipating this, but it was a really good lesson. Anyone here had taken soils class? I hadn't taken a soils class, so this was really steep learning curve for me when I started doing this work. What happens when you, yeah, good. All right. So when you shift pH, soils that naturally have a certain, they stabilize at a certain pH based on the mother materials that the soil's made of and based on things like rainfall. So we have a high rainfall area, so we get constant acidification of the soil every rainy period. When you shift soil pH unnaturally by, for example, putting in calcium carbonate, it shifts the balance of relationships among all these nutrients in the soil. And many of the micronutrients become unavailable. They're still there, they don't disappear, but they get tied up. And micronutrients are essential in micro quantities, but plants need them. And ironically, so does fusarium. And this is part of the suppression. So as you make these micronutrients less available to the host, to the fungus, that's why you get less disease and you get better yield. But at some point, it becomes critical for the host too. So we've shown that if you shift the pH enough, if you've got very acid soils and you're doing a lot of liming, if you get too extreme in that, you may have to balance this with a foliar feed of these micronutrients. You want to deprive the pathogen in the soil. So clubroot on brassicas is another example. You raise the pH because it does well in acid soils. At some point, you have to be careful that you're not depriving the host as well as the pathogen through that. So that's all related to micronutrient availability. Okay. And we had a PhD student who did a whole series of trials showing how you can really, if you look at that picture on the right, look at the difference in yield. There's 3 different female lines she was testing. But look at how much we can increase our yield by optimizing the way we amend the soil with limestone. So I'm not going to get into the details. I'm happy to discuss this if anyone wants to know more about liming and suppressing of certain soil-borne diseases. But it's a very good, sustainable way to manage certain soil-borne diseases that are quite recalcitrant if it's done carefully. All right. So switching on to irrigation practices. Do we want to stop and give people a chance to stand up and get oxygen in the brain? I can see a few eyelids getting heavy. Why don't we take 5 minutes and move around, because our brains can only take so much without oxygen. Is that okay? Just a few minutes. Sorry for the folks online, but they can do the same thing, yeah. That's why I tried to.
I forgot. I have a little bit of Okay, folks, we're just gonna, if you want to grab a broom or anything real quick, otherwise just come and take your seats again. If you've had a chance to stretch or so. Okay, I didn't mean to cut off that quickly. So just be winding down your conversations and finding your seats again so we don't lose that stretch of time. But I think that was a good idea.
Yeah, I could just, it's hard to sit. Yeah, it's hard to,
Yeah.
I wouldn't be able to sit. Need oxygen.
Okay, so everyone's gotten what they need to get and had a little stretch. We'll come back to our seats.
Okay, so I'm gonna just start talking because the online folks are also needing to get through this, but I'm gonna switch to irrigation. And you know, if you're in a climate like this part on the west side of the Cascades, you might not need to worry as much about irrigation. If you're in, you know, Penticton or east of the mountains, irrigation is critical. You don't have a crop without access to water. But the way you manage water in your crop can make a big difference on diseases. Because particularly the fungal and bacterial pathogens are very heavily moisture driven. Without moisture, these organisms don't spread or don't do very well. There's some exceptions, like in everything, but this is a very good thing to think about when it comes to the fungi and bacteria. If you are irrigating and have that capacity to use something like drip versus overhead, it can make a big difference in disease pressure. And I realize there's lots more to consider in whether or not you can afford it or it's logistical and so on, but that can make a big difference. And the timing of your irrigation can be very important. Why do you think timing of irrigation could be important? Obviously, you irrigate when your crop needs it. So that's, you know, I'm not, but time of day. Why would time of day of irrigation be important?
Well, just like if you irrigate at like 5 o'clock, then you're not considering planting with all day.
Yeah, so with these fungal and bacterial pathogens, the duration that your canopy stays wet is going to determine the amount of disease pressure to a large degree. So that if you have to irrigate with overhead, and you can irrigate in the morning so that your crop will dry out quicker before you get into the cooler nights, you're going to reduce that duration that your canopy stays wet. So absolutely, yeah. All right. So going back to that carrot xanthomonas situation, I just want to give an illustration. We were surveying for this pathogen when they were trying to find out how bad it is. And we had these pairs of fields. So for example, the OM, there's M with overhead irrigation, M with drip. There's N with overhead, the O stands for Oregon. So we did Oregon, Washington. But you can see in almost all of these paired cases, the drip irrigated part of the field had far less xanthomonas on the harvested seed than the overhead irrigated, because it's a splash dispersed pathogen. And there's a few exceptions there, but this is just monitoring the disease over time in these carrot seed fields. The higher the bar, the more disease, the more pathogen we were picking up. And the drip irrigated part of those fields always had less, with one exception. Does that make sense? I'm kind of rushing through this because of time, but so planting practices. Can you adjust your planting date? Sometimes that's not an option because you have to plant sequentially to fill your market, your CSA or your farmer's market or whatever it might be. But sometimes you can adjust your planting date to be less favorable for a particular disease. For example, if there's insect vectors that only arrive in your area at a certain time of year, can you get the crop planted early enough that you escape, or your crop gets big enough before the insect comes in and potentially carrying that? Let's see, I'll give you some examples here, but very important on planting practices is your row spacing and your plant spacing and your row orientation. And I know that sometimes that's constricted by the size of your ground, the size of the wheel spacing on your tractor or whatever your equipment might be, but I'll give you some examples. So, and this ties back to the whole idea of airflow and air movement through your canopy and the ability for that canopy to dry out, okay? So this, kind of going back to planting date, this is a picture I took in a buckwheat seed crop in central Washington. buckwheat is now growing quite widely in central Washington. It's a relatively new crop to that area. And when they started seeing problems show up, it turned out that beet curly top virus, a virus that is carried by leafhoppers, was having a devastating impact in buckwheat seed crops that were planted late. Because the leafhoppers their population builds once the temperatures warm up in the summer. They like warm and dry. So early in the spring, there's very low leafhopper numbers. If you can plant earlier in the season, you can get your crop big enough so that when the leafhoppers come, if they're carrying the virus, there's less damage to your crop. It's not seed-borne, so that's not the issue, but it can devastate yield of your crop, okay? These are pictures of cabbage seed crops, and we learned this lesson very, very well the first year that I had to do some cabbage seed crop trials about good airflow through your canopy. If you can tie up your cropping and orient your rows into the primary wind direction, if you have a primary wind direction on your farm or in your area where you're farming, and you can take advantage of orienting your rows into their primary wind direction, it can make a huge difference on disease pressure. We saw this. I had a trial. This is a trial we planted. It was the first year I was growing. I wanted to do a good job and show the seed growers I knew what we were doing. And we tied and staked like crazy. And we inoculated with this pathogen 3 times. This was on our research center, trying to get enough disease pressure. The farmer nearby had the exact same cabbage seed crop. He planted perpendicular to my direction. He didn't stake and tie very well. And he had 5 times as much disease as I had. And I was inoculating. So it really illustrated for me the degree to which managing your canopy can have a big impact on disease pressure. And so this is the disease that we were dealing with, alternaria. It can get on the seed. There's a very, very strong effect of this pathogen if it's on your seed on germination. Your germination will drop if your seed becomes infected. So very important to manage this.
Do you ever get into any, like, with brassica also, do you ever get into any pruning?
You certainly could, but anytime you prune, you're removing flowers, which is potentially seed. So that's one of the reasons that growers stake and tie. They come through with baling twine and stake and tie the racemes up so that they're more upright versus falling over. Certain parent lines are much more leggy than others, but it's labor every time you have to come through. And so that's, the balance you have to think about. But if you prune racemes, you're pruning flowers, which means pruning pods out. Yeah. So.
If you're baling and twining everything in, are you kind of compressing the canopy?
Absolutely. So you have to think about that as well. But if you can get airflow through the canopy, it's at some point you're getting more airflow through the canopy than if you're just letting it lie. And these things can get really bushy. I don't know if you've grown cabbage seed, but depending on, especially the open pollinated types that most of you are likely to be doing, they tend to get really bushy. And if you can't even walk through the rows, you're not getting airflow through the rows. So, but yes, certainly within there you can have issues. All right, so transplanting and hygiene, thinking about if you're doing plants that have to be transplanted, any injury to those transplants, wetting those transplants, handling them, potential to move stuff around as you're handling them, sanitation. So if you see problems in your transplant operation or the nurseries you're getting your transplants from, sanitation and roguing out suspect plants at that stage is really important because you don't want to propagate. Some things can be mechanically transmitted. So if you're handling things, one of the worst cases is tobacco mosaic virus. If you have smokers, if you're a smoker or you have smokers working for you, TMV will survive the flu curing process with tobacco and your workers will have TMV on their hands. So it's just something to think about. Now, TMV is primarily a problem on solanaceous species like peppers and tomatoes and potato and so on, but it's very easy. If someone needs to take a smoking break, all they need to do is wash their hands with warm soapy water, and it'll kill the virus. But you should not be handling plants immediately after you smoked if you haven't washed your hands. Okay. So I'm not going to spend a lot of time on chemicals, but I know that just because you're organic doesn't mean you don't use chemicals. And people say, well, organic is pesticide free. Nope, there's organic pesticides. There's organically certified coppers, there's sulfurs, there's things that are used in organic agriculture. It's just whether or not they fit into that category of synthetic versus naturally occurring. So these principles still apply, and there are a lot of things used in organic agriculture. You may have a philosophy of, I don't put anything on my crop besides fertilizer. There's a wide range of what is defined as organic. So I have to do a lot of work with organic growers on this principle of using pesticides, organic pesticides, sustainably and more environmentally sound systems. But there's a wide range of things. And I see a huge amount of inappropriate use of chemicals, both in conventional and organic ag. Not deliberate violations of laws, but poor understanding of how these chemicals work, both on the organic side and the conventional side. And we're all guilty of that to some degree because there's so much knowledge out there that we don't have time to understand everything and the chemistry behind everything. But I've sometimes been shocked at the degree to which, particularly in conventional, because they tend to be very chemical heavy, the lack of full understanding of how these products work well and how they don't work well, and the misunderstanding of what a word means on the label. And so I can tell you some horror stories over the years. And it's not horror in terms of, again, people being very illegal and violating major, it's just not understanding very well. And you see this in human medicine, abuse of medicine, abuse inadvertently, not recognizing how some medicines can impact things. But if you are using any kind of sprays, whether it be including organic, some important things to consider is, are you using them at the right time? Are you using them in the right way? Are you getting good coverage? Because those can all have a big impact on whether or not your applications are doing anything. Okay, so I won't go into detail, but you have the slide there. One example I have here is we did a series of trials with this disease called white mold that goes to many, many different crops. It goes to brassicas, goes to carrots, it goes to lots of species. We did a whole bunch of fungicides, including some organic. So the two in organic are circled in green because sometimes growers in organic production wanted to know if there's anything they could use. So this was Serenade, which is a bacterium, Bacillus subtilis, and Contans, which is a fungus that feeds on the white mold fungus, combined with Serenade. And growers had been using the combination of Contans and Serenade and said, is it really working? And we looked at it with and without the Contans, and you can see we got better control without the Contans. And I won't get into details, and we can have more discussions later, but again, sometimes you're spending money on stuff that may or may not be helping you. And I see huge problems in organic production because there's a tremendous amount of snake oils on the market. And I call them snake oils because there's a lot of used car salespeople out there pushing these products with all these wonderful claims of, oh, they're growth promoting and they're, you know, they're healthy for your soil and they're going to replace the bad guys. And most growers are wanting to be environmentally sound and take on these more biological based approaches to managing these diseases. And I think there's a huge abuse in the market. And I think most organic growers would be better off not buying these products and just relying on their own natural biology, building a healthy, naturally occurring microbial population, than putting in these microbes that 99% of them are a waste of money. And I've probably spent $500,000 evaluating biological products in various vegetable disease management systems. And almost nothing works like the labels say it does. And I'm not saying it's because I don't think you should use stuff, but I've been horrified at the abuse of the market, because for biologicals, there's very, very poor standards. No one has to meet these standards of proving efficacy. You don't have to prove a product works on something to put that something on the label, which is, to me, unethical, but that's the rules. I don't know about Canada, but certainly in the US, you have to prove that it doesn't hurt the crop, but you don't have to prove it works, and you can say it works on something even if you haven't proven it. So there's a lot of misinformation on these labels, unfortunately. We can go into lots of stories around that. One of the things I run into a lot, I won't go into too much detail, but this is one of the most egregious issues I've run into, is some labels will say this is systemic. What does systemic mean to you if you see systemic on the label? So it's going to move throughout the whole plant. Okay. All right. That's what I would think too. Systemic usually means taken up and moved around. Unfortunately, there's different forms of systemic movement. So there's a thing called translaminar. Translaminar is something that you apply it on one side of the leaf. It'll be taken up into the leaf, and it'll move across the leaf, but it won't move down the plant. It won't move other than the leaf it's put in contact with. You don't have to worry about getting on the lower surface. If you can put it on the top surface, it'll protect the lower surface too. So it's very limited systemic movement. Many products are sold as systemic that are only translaminar. And so you might think, oh, I don't have to worry about good coverage because it says systemic on the label. But if it's translaminar, it's not moving anywhere except across the surface of it, from the top surface to the lower surface of that leaf. A lot of products are locally systemic, so they might move, you know, a limited degree from the bottom of the leaf to the top of the leaf or so on. Most products that are systemic only move when it comes to diseases. I'm not talking about herbicides, I'm not talking about insecticides, because those are quite different. But with fungal and bacterial type products, when they talk about locally systemic, they're moved in the xylem. So which way does xylem move water? Up or down? Xylem does not move water down. If something's moved in the phloem, which way does phloem move photosynthase? Up and down. So phloem can actually transfer photosynthase up and down the plant. So products that are moved in the phloem will move up and down a plant. Products that are moved in the xylem will only move one way. So I see growers look at the label, they say it's systemic. They may have a problem with, say, Pythium leek on potatoes and apply it to the canopy thinking the plant's going to move it down to the roots. It's xylem translocated. It's never going to go down to the roots and to the tubers, never. And yet it's being used in that way by many growers. So understanding this, when you talk about systemic, and it's to the benefit of the companies to not explain this on the label, because they can sell more. Sorry, but I just see this happening all the time, and it's a real tragedy. So you had your hand up? Yeah, how do you find out? Okay, really good question. So if you, and this tends to be primarily pertinent for synthetics. So fewer, not very many organic products have systemic movement. So you probably have less issue to resolve this, but it's a huge, huge problem in conventional production. Herbicides and insecticides, most of them, when they say systemic, they are phloem translocated, but with fungicides, it's almost all xylem translocated. There's only one family that's phloem translocated, and it's not organic. So if you see an organic product that says systemic, you know it's not going to be phloem moved. It's going to be xylem translocated. So this is why coverage becomes so important. If you are putting on a sulfur or a copper, those products aren't even systemic. They're protectant. They don't translocate in the plant. They're purely on the surface. So coverage becomes really, really important. It will not move through the plant. Okay. So this was a trial we did with carrot seed production on Xanthomonas. We evaluated a number of products, compost teas because of the interest in organic. We had a bunch of coppers because of different coppers out there. We put them on through a backpack spray. We put them on through the sprinkler, mitigating chemigation. The best control we got, the lowest bar, was chemigated. So why would chemigation, putting on through the sprinkler, do better than putting on with a spray boom on a tractor or a backpack spray? It's coverage. So your water's the water is what's dispersing the Xanthomonas, but it's carrying the product with the water that is your dispersal agent. So coverage is really, really important. Okay, I'm going to move on because of time, but this is one of those leaf spot pathogens I talked about on spinach. Here you can see it developing in a seed crop. This is a commercial seed crop we were looking at. You can see it starting to form on the seed. One of the things we found out with one of these two pathogens, so there's Cladosporium and Stymphalium, if we put pollen on the plants and then add the fungus, we get 5 times more disease. Why do you think that is? Okay, it's a food effect. So Stymphalium feeds on the pollen. So if you're a seed producer and you have to have pollen because it's a wind-pollinated species, it has to be cross-pollinated. You know that Stymphalium leaf spot, the disease pressure is going to be greatest when your crop is starting to shed pollen. So if you do need to put on a protectant, this is the time, just before you start getting into the anthesis, this is the time you want to protect your spinach seed crop. And if it's dry, you don't even have to worry about it because it's moisture-driven disease. But pollen is a food source for some pathogens and not others. So you can see Cladosporium wasn't affected by the pollen, but Stymphalium was 5 times more aggressive when there was pollen on the crop. Okay, I'm going to, for onions, this is a disease, I mentioned botrytis, the gray mold fungus that affects onions. When it comes into an onion seed crop, this is what happens to your umbels. This is what gets on the flowers, it gets on the seed, and then when you plant that seed out, potentially you have bulb rots. So this is a picture I took in South Africa. I was working with some onion seed growers, and one of the growers said to me, I never seem to be able to get enough nitrogen in my crop. It always turns bright yellow. This was not nitrogen deficiency at all. So if you look at the base of these plants, as onions come out of the winter and they bolt and they push out that seed stalk, the leaves at the bottom of that plant die around the base of the stalk. The stalk is still alive, but the leaves die. If you do overhead irrigation or you have a lot of rain, water gets trapped between that sheath of dry leaves and the seed stalk, and botrytis will grow inside there. And so I went and showed the grower, I pulled the sheath of dry leaves back, and you can see those little gray rings. That's all the botrytis, the gray mold, producing spores underneath that sheath of dry leaves. So it was basically girdling the seed stalk, the scape. And so the plant was continuing to try and make seed, and it took all the nitrogen out of the scapes to feed that seed. So it looked like it was nitrogen deficient. And the grower was just dumping nitrogen on as best he could. And he was just exacerbating the problem. And it's all about the use of overhead irrigation during some critical periods when that leaf sheath was dying and trapping the gray mold underneath it. Okay. The other period that we see problems is, this is where we see gray mold attacking the umbel where it attaches to the scape. The reason is when the umbel opens up, that membranous sheath that surrounds the umbel, it opens up and it stays attached to the scape. Now you've got a cup-shaped layer of dead plant tissue, and if you're overhead irrigating, you're putting this nice cup of moisture attached to the flowers for the gray mold to grow, and it just grows in and it kills the scape where, right at the point of attachment of the umbel. So being conscious of this, if you're irrigating and recognizing these risky periods, can make a huge difference in your ability to manage these crops. Yes, it is, unfortunately. Yeah, yeah.
So a lot of this could be controlled just by using drip irrigation?
Yep, absolutely. And now if you're in a wet climate, no. But if you're in a dry climate, that's the beauty of growing. That's why these seed crops are grown in drier climates, is that you have more capacity to manage these diseases culturally. Yeah.
So are most seed growers in this area using drip?
Well, some of the conventional growers will rely on chemicals to make up for that difference, but organic growers, a lot of them are shifting to drip. So in Central Oregon, because of the xanthomonas problem, about 60% of carrot seed production is now drip irrigated. When I started in 2000, it was about 10%. It's all because of xanthomonas. So it works. Yeah, and it's great. You don't have to rely on chemicals. Yeah, but if you're in a wet climate, you don't have a lot of control over that. Yeah. I'm going to show you a picture of, so this is not a black and white picture of carrots. This is powdery mildew. Oh, sorry, you had a question?
So the traffic seed and soil.
It's soil-borne to a limited degree. It'll form these structures called sclerotia that are about mouse turd size, mouse poop size. And they'll survive in the soil for a couple of years, but eventually they'll be colonized by other fungi and start to break down.
So if you had an outbreak at the level of
well, in that particular field there's a lot of inoculum. So what I would have recommended to him is once he harvested a seed crop, is to go ahead and get that residue worked in the ground and stay out of that field for about preferably 5 years. And most seed crops are on a small scale compared to a bulb crop, for example. So you have more options for rotation. And most seed growers tend to be out of a field for at least 3 to 4 years, preferably 5, because if you have seed that's shattered and landed in that, and those seed volunteer the next year, you don't want cross-pollination if you're growing a different variety. So seed growers tend to be good on rotation. So that's a beauty of their disease management plans. Yeah.
So he would have still harvested the seed from that field?
Yeah, he did. Yeah.
And then would it be treated so it's not?
So what he could do there is test it and see, and you can if you're a conventional grower, there's some fungicides you can use. If you're an organic grower, you can look at hot water seed treatment. It's pretty risky with onion seed. People don't like to wet onion seed. It has a limited shelf life as it is with onion seed. So, but yeah, for conventional growers, there are some fungicides they can put on the seed. Yeah, yeah. So this is a picture of powdery mildew on carrots. There's some really good resistance to powdery mildew, but if you happen to be asked to grow a seed crop of something that's super susceptible, this is unfortunately what can happen because of the dense canopy. The picture on the right is from a trial I did where we would grow carrots of a very susceptible variety in the greenhouse, get them colonized by powdery mildew and transplant them into our trials as inoculation. That's how we, and I mean, they look like little albinos in the field, but that's all powdery mildew. And what we wanted to see is the growers were saying, well, is powdery mildew important? Is it affecting our yield? Do we have to worry about managing it? It's not a big issue in terms of seed-borne inoculum, but is it actually damaging crop? So in these trials, we inoculated the plots at different times through the season. So this is the second year of the biennial season. We inoculated in April, or we waited to inoculate till May or June. But what you can see is this is the severity of powdery mildew on the y-axis. So you can see in the beginning, it didn't matter if we inoculated in April, May, or June, the disease didn't take off. Why? What drives powdery mildew? Humidity. So you've got these little carrot plants coming out of the winter, and your row spacing is quite wide because you're going to get a crop that's, you know, this tall. And there's no canopy closure. There's air movement through the row. There's no buildup of humidity. This is in central Washington. It's dry. As soon as canopy closure occurred, so your crop got tall enough, the rows were closed because your crop's now this tall. Look what happens to the disease. It just skyrockets. Whether we inoculate in April, May, or June, the disease didn't take off until canopy closure. And then if we inoculate in July, it very quickly took off, caught up, and you can see versus when we inoculate in August, or if we didn't inoculate. And what happened to seed yield? This is the marketable seed yield we harvested off these plots. So if we inoculated April, May, or June, it didn't matter when you inoculated, ultimately we had the same yield, about 1,500 pounds per acre. If we kept that disease out of the field until July, we increased our seed yield 20%. If we kept the disease out till August, we increased our yield 44%. And that translated into between $240 and $700 per acre difference in price to the grower. That's pretty substantial. So managing diseases can have a big impact. That's open pollinated seed when it was selling for about $1.55 per pound of seed. This was the numbers. This was about 8-9 years ago. Okay, so we talked about resistance. It can be very effective. There's some excellent resistance to powdery mildew. If you aren't being dictated which variety to grow, looking for a resistant variety can be really important. Unfortunately, a lot of seed growers, particularly on the commercial side, have no choice. They get a contract, you will grow a parent's XY because we want hybrid Z, you know. In the world you work in, that's the beauty of your system. You have a lot more choice. You're usually not being dictated to quite the degree that these commercial seed growers have to deal with. This is a picture I took last year of a spinach seed crop in Skagit County. It's a hybrid, you can tell, because there's male and female rows. This is a postdoc and a graduate student in my program. This field had been out of spinach for 18 years. That's Fusarium wilt. So that disease persisted in that field for 18 years. The male was quite resistant. See, it survived. The female was very susceptible. That grower had no choice. Those were the parents she was asked to grow. She had a 40-acre field. Her yield was reduced 75%. Now, she harvested some seed off that. It didn't totally kill it, but it wasn't economically viable for her. And she said to me, Lindsey, I would have planted the crop in the field next door if I'd known that the risk was so high. 18 years out of spinach should have been safe enough. So over the years, we've developed a bioassay that this year will be our 11th year of offering this bioassay to spinach seed growers because this disease is so persistent in our soils. And every year the growers bring us a 5-gallon bucket of soil. We plant it out with spinach lines that range from super susceptible to partially resistant. This is a field that everything died in. That was a field growers considering planting. It would have been very similar to the female line that grower last year had. This is an intermediate risk field, and this is a safe field. And we've done this, we've tested over 405 fields now over the last 10 years for growers in Western Washington, showing the level of risk. The higher the bar, the more risk for that soil and very much dependent on the parent line, how susceptible your parent line is. This is the first year we did the test. This was the next year we did the test. It was so successful, we ended up with 50-something fields submitted that next year. We charged $200 a field because it's pretty tedious and expensive to run this, but it's been extremely effective at having growers avoid these wipeout situations because you would think 18 years would be enough. But if your parents are susceptible enough, and if either one dies, you don't have hybrid seed. They both have to survive. So this has been a big problem for our growers, but we also offer testing for seed companies to know how susceptible their parent lines are, because that's part of your risk. It's not just how much disease is in the field, but how susceptible their parent is. So we do this as a service, so our seed growers don't end up with these disasters that we've seen way too many times.
So on the right side, you've got a couple of.
Those are our controls.
Okay.
Yeah, we set up controls to make sure we're testing things correctly. We always have control soils with 3 different levels of risk, so we can gauge if we've done something. So say that again. Yeah, so it's partial resistance. There's no complete immunity in spinach to this disease. Yeah, we create these different disease. We grow the fungus up and inoculate the soil and create these disease pressure levels as a check that we're using. Our methods are working for the growers. All right, so if you are concerned that you have something on your seed, let's say I've got 5 minutes, I'll finish this up. What can you do about that? And there are different ways you can treat seed. Some pathogens can be treated effectively on seed and the inoculum dropped or eradicated. Others, it's not going to work. So one of the most common methods that is used quite widely, particularly for organic markers, is physical treatment of seed, particularly hot water. Unfortunately, some species cannot be hot water treated. So arugula, you cannot hot water treat. Radish seed, you cannot hot water treat. It just doesn't, it kills the seed. It damages the seed too much, or it turns into slime, depending on the species. But there are a lot of species that do very well for hot water treatment. Larger seeded vegetables, larger seeded crops this tends not to be very effective because you damage the seed too much, okay? There's a lot of interest in looking at steaming versus hot water. And the reason is when you steam, you're not wetting that seed so much, so you don't have as much imbibing. It's usually a shorter duration. So High Mowing Seeds is an organic seed company in Vermont. It's developed a proprietary steam treatment that we've tested on brassicas and spinach. It's been quite effective. We're looking at a number of other species, but again, it's not scaled up to some of the amounts and the volumes that might need to be treated, but there's a lot of promise in that. You can also look at disinfectants. Disinfectants work for inoculum that's on the surface of the seed or just on the seed coat, but if it's internal in the embryo, those disinfectants have limited ability to clean that seed up. Okay, so that's something to keep in mind. And so just, you know, the efficacy of these seed treatments is largely influenced by how deep seeded your infection might be, how heat tolerant your pathogen might be. Some pathogens are much more heat tolerant than others if it's heat you're dealing with. So there's a lot of complexity that goes into making seed treatments work. It's not simple. So if you're going to treat seed, be really careful because you can kill your seed really fast. Okay. You know, the thresholds for seed-borne inoculum, if you're dealing with brassicas in commercial trade, if 1 seed out of 30,000 tests positive, you cannot sell that seed. You have to treat it and retest it. Yeah, so the question is about biological seed treatments. There's a tremendous amount of biological seed treatments out there. And there's trichodermas, there's pseudomonads, there's bacillus species. And I can send you some papers we've evaluated some of these. And there's multiple species. There's, we don't develop biologicals in my program, but we evaluate biologicals on behalf of the seed industry that are being sold out there. There's Trichoderma hausianum is an old one. There's a number of species out there. Yep, Echeverridae is one of them. Yep. And I think one of the difficulties is with biologicals is we treat seed with these biologicals and then we put them in these very disparate environments. So you may be a grower in central Okanagan area, you may be a grower in Western Washington, and we expect the same species to survive in the same environment, different pHs, different moistures, different temperatures, and they don't always fit in those niches, those environments into which we place these organisms. So this is the difficulty, is inconsistency in performance of biologicals is one of the biggest limitations of it. They may be a niche environment in which it does well, but as a grower, you don't get to choose your environment, it's what you have. And so this is one of the difficulties we're faced with biologicals. And not to dismiss them, but there's limits that we have to recognize. Yeah. Okay. So for some organisms like Xanthomonas and carrots, there was a study in California showing you can tolerate 10,000 colonies of Xanthomonas on carrot seed before you get any yield loss. So they don't worry if you have some Xanthomonas on carrot seed, but if you're a cabbage grower, it's a zero tolerance disease. So different pathogens come with different risk. That's the key thing to remember here. And this is because the risk for seed transmission is influenced by so many things. If you have infected seed and you put it out in your environment, these all the things are going to influence the risk of that organism being transmitted and causing a loss. And we don't have time to go through that, but I'm gonna move through this and get to the end part here. Sorry, I know this is long, but there's a bunch of biologicals that we were looking at on pea seed for controlling damping off. And the best treatment in this case, unfortunately, was Nordox, which is an organic copper, and priming the seed. Why would that work at controlling damping off? You get it out of the ground quickly. These damping off pathogens are only a problem in the young succulent seedling stage. If you can get your seed up and out of the ground fast, you can escape a lot of pathogens like this. Okay, so sometimes on a small scale, you may have that choice, but if you're a big grower, you're not going to be able to prime all that seed. But certainly in this case, we looked at a wide range. There's trichodermas in there, there's bacillus in there, there's all kinds of biologicals in there, and really those are the only two that gave management under those conditions. Okay, these are some seed treatments we looked at on spinach, a wide range of conventional and organic. The threshold was 10% because that's what Mexico required to buy seed from the US. And you can see a number of treatments, both organic and conventional, were really effective. The best organic ones were steam treatments. Again, because of knocking back the population on this, of this case, verticillium on the seed. But just to illustrate, just because something is biological doesn't mean it works. You really have to know the efficacy. Okay. One of the really important things I think that is a beauty for folks who are doing organic agriculture is you tend to work really hard at building soil health. And what I want to illustrate here, there's more data here than you care to look at, but we planted out seeds. So if you look at the black bar on the left, that's in Mount Vernon. The height of the bar means the amount of seed transmission. So when we planted non-treated seed in non-fumigated soil versus fumigated soil, we got a 5-fold increase in seed transmission when we put infected seed into fumigated ground. Why is that? No competition. So verticillium is a very, very poor competitor. And if you kill out all the biological activity in your soil, and then put inoculum in that ground on your seed, that's one of the worst things you can do. So fumigation can have very, very nasty kickback effects. And it's building your soil health, building your natural soil microflora that is competitive against pathogens is one of the best things you can do to help with some of these pathogens. And we showed this in Washington. We did a number of years in California as well, showing that the widespread use of fumigation that was happening in areas like Salinas was exacerbating problems with these seed-borne pathogens that were transmitted. And that's really all we were getting into. Now, I'll wrap this up with seed health testing. And I think the woman who runs the seed lab here, she's just stepped out, but I'm very interested to go to her lab. But where do you get your seed tested? Does anyone here ever get their seed tested? No, this is not an arrogant question. This is a very serious question because unfortunately we have a dire shortage of seed testing facilities that have pathology expertise. And within the world of plant pathology that I work in, there is one seed pathology course offered in North America at Iowa State University. That's it. No one else in North America offers seed pathology as a course. Isn't that devastating? So one of my goals when I get rid of some of the responsibilities I have over the next 4 years is to maybe consider how we can make seed pathology curriculum much more widely available, because seeds are a foundational part of what we do in agriculture, and yet we don't have the expertise and resources available on that scale. It's pretty shocking. So do you know of seed testing facilities in Canada in this area? Okay. Okay. All right. Yeah. Yeah. So on the vegetable side of things, the 3 labs that I've historically used, they're in the US side of the border. They probably take stuff from Canada too, but there's one in California called California Seed and Plant Lab. Very good. There's one in Colorado called Eurofin's STA Labs. And there's one at Iowa State University, run by a seed science center by Iowa State University. Those are the three that on the vegetable side of things have pretty wide spectrum of tests available. You had a question, Emma?
How accessible are these labs in terms of cost? Because I know that even in Canada, when there are labs available, it's costly.
Yeah, and this is one of the big problems is these tests are not easy to run. You have to have people who are trained as mycologists or bacteriologists. So it's not like you can just take someone with minimal training and turn them into seed pathologists. And so the tests take resources, so they're not cheap. And that is one of the biggest restrictions about seed health and seed health, people getting seed lots tested. It's a huge problem.
We show them a lot because of plate approaches.
Yeah.
Or PCR techniques taking over.
So really, really important question. I have pictures here of spinach seed and onion seed plated out, old scale, microscopic examination. We still do that. So question is, why aren't we doing more DNA-based testing? So there is a lot of DNA-based testing, but one of the problems is, let's say you take a seed lot and you hot water treat it, and then you test it again. Are you going to pick that DNA up? Yep. If you've killed that pathogen, you're still going to pick the DNA up. So that DNA test it's meaningless if you don't know if it's viable or not. So this is one of the biggest limitations behind DNA testing, is the ability to separate, are you picking up DNA of a live organism or something you've killed or that was non-viable to begin with. So if you look at the National Seed Health System and the International Seed Health Initiative, the seed health tests that have been certified for international seed trade, very few of them are DNA based unless it's viruses, because of this importance of knowing if it's biologically viable or not. And most DNA testing protocols cannot tell you that. So old school mycology is still very relevant. Old school bacteriology, yeah. So there's RNA testing. A lot of it's virus many viruses, plant infecting viruses are RNA based. So there's a lot of RNA testing in that capacity. When it comes to the fungal and bacterial, there isn't a lot of RNA testing available that's been validated on the level needed for confidence in internationally standardized testing. But that's ultimately, there's a number of things that are being looked at. One is looking at RNA, which is basically saying your DNA is being transcribed. So if you're picking up RNA, you know it's active. Another one is these DNA binding dyes that separate DNA from live versus dead cells. They work with bacterial tests, but they don't work with fungal because of this complex wall of fungi. We're getting into way too much technology on seed pathology, but there are these issues that haven't been dealt with in seed testing, and the needs of the industry are so much bigger than so far what seed testing has been able to meet, unfortunately. Yeah.
We are starting to run into the lunch session.
Okay.
So we're told to wrap it up. I don't know if you're
We pretty much, this is just showing some resources where people can find information on seed. It's kind of big picture. It's not specific to a region. And with that, just thinking about monitoring your crops as seed growers in terms of seed exchange, things to keep thinking about, finding resources you can turn to if you have questions or you have uncertainties about a problem. I don't know what you have here in BC for pathology support. Vipin Goshi, I think, is someone that you can turn to. She's good. Yeah. Yep.
We're pretty supportive too. They have limited staff, but
yeah.
If you have a problem, sometimes they can come up with that.
Oh, wonderful. Yeah. Yeah, exactly. Well, that's good because that can make a big difference. All right. So thank you.
So we, obviously so important and so interesting. I think we could have gone on much longer. No, it's all right. I wasn't really engaged for time either because I was fascinated. We have run into lunch. We don't have really time for questions, which I think we all asked some questions
but
during.
I'll be around.
But
yeah.
you'll be around through the afternoon. Are you going to be here this evening?
I'll
as
be
well?
here till about 3:30.
Okay.
Yeah.
So I really encourage you to catch Lindsey and ask her your questions. Even if some of you have questions as a group, you might want to find her in a corner and set up your own little discussion. And we have, Lindsey is an advisor to the CANOVI project, so we have, we can contact her with other questions down the road and continue the conversation. So thanks
Thank you.
again.
Thank you.
This is great.
Thanks for the opportunity. I hope it was helpful and not too upsetting. Thank you.
Thank you so much. No, I don't know how to work this. Yeah. But it makes me think that I think this is a good start to get everybody. Well, we can just end for laughing around. That will stop a lot more. Formulate more questions.
I think we want to make sure we do it well. So it
Yeah, well,
does-
stop hogging your time.
Oh, good. That's my job.